Hi, my name is Prajwal Paudel, Product Development Scientist at ACROBiosystems, and today I will present ACRO's effort in developing critical need reagents and assay kits to fight COVID-19 and SARS-CoV-2 variants. The COVID-19 pandemic has infected 112 million people worldwide, with 2.5 million deaths to date. The viral agent of this pandemic, novel coronavirus SARS-CoV-2, belongs to the beta coronavirus family clade B, which includes the SARS-CoV and MERS-CoV outbreak from 2002 and 2012. SARS-CoV-2 viral particle consists of a single-strand positive sense mRNA wrapped around a nucleocapsid or the N protein, and this genetic material is enclosed by a lipid membrane decorated with the membrane protein and the envelope protein. SARS-CoV-2, as with other coronaviruses, are defined by the projection of spike glycoprotein trimers. ACRO has developed a variety of high-quality SARS-CoV-2 proteins to facilitate the diagnostic and therapeutic development against COVID-19. Our catalog contains the three main SARS-CoV-2 structural protein, the S, E, and the N protein. The S protein is primarily expressed in the HEC293 cells and includes his tag and biotinylated ABI tag. The N protein is expressed in both E. coli and HEC293 cells. Due to high number of N protein present during infection, antigen-based diagnostic utilizing N protein detection has been mobilized during this pandemic. E protein is the smallest structural protein of SARS-CoV-2, and we provided it expressed in E. coli. In addition, we also offer the non-structural proteins that are all expressed in E. coli with his tags. SARS-CoV-2 spike glycoprotein is a fusion protein homotrimer comprised of two subunits. The S1 subunit contains the N-terminal domain and the receptor binding domain that binds to the ACE2. The S1 subunit is a dynamic structure with a closed and open conformation. The open conformation binds tightly to the ACE2 receptor. The S2 subunit comprises of the fusion peptide, heptad repeats, transmembrane region, as well as cytoplasmic domain. In the trimer form, the S1 forms the head, supported by a central stalk comprised of the S2 subunit. Prior SARS-CoV research had already established the S protein to be highly immunogenic and key to viral infection and propagation, allowing for faster therapeutic and vaccine development against SARS-CoV-2. ACRO provides S1 subunit and S2 subunit, as well as NTD, RBD, and CTD domains with biotinylated ABI tags, NFC tags for a wide range of detection and diagnostic assays. To generate vaccine and therapeutic antibodies, pre-fusion stable S protein with high recombinant protein expression yield and homogeneous preparations are needed. 6 proline substitutions have been identified to stabilize loops and cap helices in the pre-fusion state. ACRO offers a high-quality Superstable S trimer, improving upon the 2 proline mutated trimer. This protein contains a T4-fibritine trimerization motif and a double alanine mutation to abolish the furin cleavage site. The S trimer has shown high stability and high homogeneity as seen in the negative stain electron microscopy and the Moll's experiment. Functional binding with H2 also shows comparable binding affinity to literature values. In addition to that, functional binding or performance comparison in antibody titer measurement also shows our Superstable S trimer performing with highest signal-to-noise ratio compared to other S trimers. ACRO's development of reagents and kits has had measurable effect in efforts in supporting rapid vaccine and therapeutic development, all the way from discovery to preclinical to clinical stages. In fact, we have helped NIH researchers identify novel protective nanobody that blocks ACE2 S protein RBD binding with high potency. Antibody-like monobodies were discovered using ACRO's catalog of S protein and ACE2 and were found to neutralize SARS-CoV-2 infection in vivo E6 cells with sub-nanomolar IC50. Additionally, ACRO's S protein RBDs were utilized by Pfizer BioNTech to assess immunogenicity of their mRNA vaccine candidate in a phase 1/2 study. Our TAS-K022 Neutralization Kit is based on competitive ELISA principle. This neutralization assay approximates the gold standard print assay that uses live viruses and cells, requiring a P3 laboratory and a complex low throughput assay. Our kit contains the immobilized ACE2 protein, which can be incubated with serum samples followed by incubation with HRP-tagged spike RBD. The signal intensity of the ELISA is inversely proportional to the presence of neutralizing antibodies. This kit is semi-quantitative and shows good dynamic range and high sensitivity with controlled neutralizing antibody sensitivity at 10 nanogram per mil and a high lot-to-lot consistency as well. In fact, we tested the neutralizing kit with vaccinated serum samples for the presence of neutralizing antibodies, and the kit shows 98.5% specificity and 100% sensitivity. In addition, comparison with the FDA-approved EUA-approved ELISA kit shows our kit having strong correlations in the antibody titer as well. ACRO'S support for diagnostic development effort constitutes of antigens and antibody detection using LFA and ELISA methods, sensitive enough for asymptomatic and symptomatic patient samples, and also for confirmation assays. In fact, we support immune response characterization with our antibody detection and antigen testing using our catalog of anti-N and anti-S antibodies. S1 protein were used to assess IgG subtype titers in hospitalized pediatric and adult patients in Sweden. And ACRO also supports a population-wide screening to assess viral spread, exposure, and vaccine efficacy. And ACRO's N protein was used to determine distribution of SARS-CoV-2 exposure in healthcare workers and showed a significant risk in direct patient contact workers. ACRO has also developed antibody match pair for lateral flow assay against nucleocapsid and spike proteins. These antibody pairs have high stability and specifically are designed for serological tests. The nucleocapsid detection pairs shown here performed extremely well with positive test line at tissue culture infectious dose value of 10. ACRO also offers highly specific and sensitive mouse and human IgGs to detect SARS-CoV-2 antigens. Here is an example of S1 specific and N protein specific antibodies that are exclusively specific to the SARS-CoV-2 spike protein compared to the other coronavirus spike protein. We also offer 11 mouse and human IgGs as well. Additionally, we also provide antibody detection kit for total anti-RBD antibody titer detection, anti-S trimer IgG titer detection, anti-RBD IgG 1, 2, 3, and 4 detection, as well as anti-IgM titer detection for RBD and S1 proteins. We also provide high-quality control RBD and NTD neutralizing antibodies to benchmark the kits. In addition to the ELISA kits, ACROBiosystems is proud to offer first-in-class S trimer, S1, and RBD protein pre-coupled magnetic beads for antibody screening and therapeutic applications. The magnetic beads reduce time and cost, as well as are convenient products for research and development. ACRO has done extensive stability testing to show low nonspecific binding and a consistent performance across multiple freeze-thaw cycles. Leading vaccines with strong efficacy against the original strain of SARS-CoV-2 has shown lowered efficacy for the South African variants, among others. These data are alarming and need to be closely monitored for natural and vaccine immune response escape to prepare against a potential surge and severity in cases. So to support this work, ACRO has developed more than 60 SARS-CoV-2 mutant products covering point mutants in S protein, NTD, and RBD regions. ACRO has also provides top variants including the UK variant, the South African, and the Brazilian variants for diagnostic, vaccine, and therapeutics development. ACRO's arsenal against COVID-19 pandemic is geared towards rapidly accelerating research and developing processes which encompasses high-quality proteins, antibodies, kits, and pre-coupled beads. Here's a summary of ACRO's full effort in diagnostic, therapeutic, and vaccine development effort against SARS-CoV-2 and COVID-19 at each step of the viral infections. ACROBiosystems has a global presence with offices in Beijing, Shanghai, West Coast, and East Coast. We have more than 5,000 customers in over 60 countries in pharma, biotech, and CRO, diagnostic, and academic space. ACROBiosystems is ISO certified, and some of our COVID-19 diagnostic kits are also IVD certified. In addition to the COVID product line, we also offer more than 1,600 recombinant protein catalogs, including immune checkpoint proteins and CAR-T targets. please visit ACROBiosystems.com for more information. You can contact me. Here's my email. For any general inquiry, you can contact order@ACROBiosystems.com. Thank you.